Welcome to Convocation today. We're glad that you've joined us. Today we're pleased to welcome Dr. Allie M. Graham to the stage. She grew up in Houston, and I'm really interested in you hearing what she has to say about her experience. She is a scientist, she is a researcher in biology, and she has a bachelor's degree, master's degree, PhD in biology, but she didn't come to it through, I guess, the traditional means. And she's going to tell you a little bit about her story, how she arrived at where she is today. And I hope that some of you who are maybe kind of wondering, oh, what do I want to do with my life? I'm not sure, what I want to study, or, am I on the right path? I hope you'll listen to her message and kind of take heart and realize that, sometimes you just don't know, exactly where you're going to land. But you just get there and you learn along the way, and eventually you will accomplish great things. She has her bachelor's degrees from Randolph-Macon Women's College, and then her Master of Science is from University of North Carolina, Greensboro, and then her PhD is from the University of Miami. Okay, she's done a couple of postdoctoral gigs. She did a National Science Foundation postdoctoral research at Oregon State University, and then she's currently a National Institutes of Health Pathway to Independence Fellow at the University of Utah. Please join me in welcoming Dr. Graham to the stage.
Is it okay now? Okay. All right, thank you so much, Cheryl, for that introduction. So it's really exciting to be here and to kind of give this type of talk. I'm normally invited to give talks to biology departments, and so the structure of my talks and what I talk about is slightly different. So I've titled this particular talk, given I know what the audience is going to be, to include obviously some of my science, but also to pepper in little bits of story about how I got to where I got to with that particular question, or even just a little bit of my literal path or journey as a scientist. So that's why I've titled my talk, the head of the talk, From the Mountains to the Sea, which references the type of research I work on, and then in quotations a little bit about me. So I'm kind of peppered in throughout there. So don't worry if maybe some of the terminology I use is, you don't really quite understand what I'm talking about, or that you zone out when you see something. It's not important that you know all the specifics, just, try and keep an open mind for the general picture. And I have individual slides every now and then that tries to summarize what I've shown you in terms of graphs and figures. So the breakdown that I have for this particular talk is a little bit about me and how I got to where I've gotten to, since obviously Cheryl, she read out a lot of different institutions all over the US. And then a bit more introduction about my specific research questions and why I think they're cool and why I think studying response to low oxygen or hypoxia is kind of important on a number of different levels. And then we'll get to some of the more interesting, research questions that involve a variety of different animals. So there's pretty pictures and stuff, so hopefully it'll keep you all entertained a little bit. So I started out just like many of you as babies. So how did I go from this wide-eyed little girl that's a first-generation college student? My parents were also in the Navy, so daughter of veterans. My mother was a stay-at-home mom, and my dad went to trade school for architecture and design. But I grew up loving being outdoors and going to science museums and such. So how did I go literally from there to Dr. Allie Graham, going to all these different places, getting a number of degrees, and then being awarded pretty prestigious fellowships from both the two major funding institutions here in the United States, the National Science Foundation and the National Institute of Health. Okay, so I alluded to my literal scientific journey throughout the United States. I was born in San Diego, where my parents were stationed in the Navy, and then after a number of years, I think I was around 5, that they left the Navy. We moved to Houston, Texas, so my whole family is pretty much in Houston, if not in the surrounding areas. And like I said, I grew up going to, going outdoors and birding, because my grandmother really liked birding, and then going to science museums and such, and loving dinosaurs and Indiana Jones, and then eventually falling in love with genetics, and then wanting to do biology, but not really having any preconceived notion as to what I would do with it. I just know that I liked it and I was interested. So I eventually went to Randolph-Macon Women's College for college in Lynchburg, Virginia, in the Blue Ridge Mountains. I ended up going there because it gave me the most scholarship money. It was a very small school and didn't have much of if any, of a research program. And I wasn't able to do any research in the lab or in the field. During the school year, I was working, and then also during the summer, which is when most other opportunities pop up, because I was also working, both as a lifeguard and retail, to support my parents and pay for school. I knew that I wanted to continue, and research sounded really exciting, and trying to answer questions was of interest to me. But I didn't really know how best to do that, and PhD programs didn't quite seem like the right route, given I felt what it took to get into those programs. So my counselors at the time suggested, well, maybe you should think about a Master's of Science. It's a terminal degree. It's only 2 years. You'll get you'll get the support, and you'll learn a whole bunch of new techniques that you can decide if you want to stop right there or if you want to move on to do something else. So that's where I eventually did my master's in biology at University of North Carolina, Greensboro. So I worked with honeybees and learned a lot of new techniques at the bench and at the computers, using computational work and bioinformatics, and then as well as learning molecular biology and genetics and genomics. And then after then, I liked the question asking portion of it, but I didn't quite fall in love with the system, and I didn't really know what I wanted to do. So I eventually, for a number of years, worked at Duke University, both as a researcher in a particular lab, and then also taught genetics and evolution for a couple of years. And then after some time really sitting with myself and figuring out the types of things I wanted to do, I applied to PhD programs, but only a handful, because it was kind of expensive to do so just a very select few. And I ended up going to the University of Miami for my PhD. I had a little bit of a rocky start. My initial lab I was in didn't quite fit, and I didn't like the PI very much, so I switched labs, ended up graduating in 4.5 years, which is very quick. And then in searching for postdoctoral research, locations, I had really sat down thinking what I wanted to do next and how my PhD research would fit into my postdoc research. And if you're not familiar with a postdoc, is kind of like also on-the-job training for you to be more independent, learn to be a more independent researcher, and come up with questions you can use to create your own lab somewhere as a faculty member. So I did that at Oregon State University, where I was a National Science Foundation Fellow. So I was on the job market and everything, and then COVID hit and really screwed everything up. So I decided to take another postdoctoral position, and as long as it continued to do the work that I wanted to do, and that's how I ended up at the University of Utah in the Human Genetics Department. So for a lot of the ways, I've obviously traveled all the way around, it seems like, and I'm not sure where I'll end up next, but hopefully the next position is one that's a permanent one where I can do research and lead a lab and teach as well. So let's see. Yeah. So let's finally kind of get to the research portion of this. My particular interest, as Cheryl mentioned, as well as the title of the talk, revolves around how organisms are able to respond to low oxygen situations. So low oxygen I will call that hypoxia. So very specifically, I wanted to include both terrestrial and aquatic systems, and I'll talk about those. So one of the best studied kind of terrestrial hypoxic situations is in the altitude. So obviously, at sea level, we're at about 21% oxygen load, but up in the mountains, even here, it's around 17 or 16%, which if you're not from here. Maybe it took a while to kind of get used to, at least that was for me as well. And some of the mountains, it gets up into the, 10 or 9, so we get pretty high. But there's so many animals which sustain populations at altitude. However, it's not without its own risks. There's abundant documentation of increased mortality and morbidity associated with settling at high altitude environments, and it comes with a really strong fitness bottleneck during embryonic development. So obviously, if you're pregnant at altitude, there's not enough oxygen. It can cause some general issues, and that can create this bottleneck for fitness. So my question was, what about the organisms that are surviving and thriving at a constant state of hypoxic stress? What are they using, both on a physiological, genetic, and molecular pathway? Because clearly high altitude situations are not completely devoid of life. There's plenty of things, and I have a variety of animals up here, both mammal, birds, even human populations, Tibet, Andes, and Ethiopia. And they're doing quite well. So along with it kind of being a very interesting question from a basic standpoint, and also part of the reason why I'm funded by the National Institute of Health, and that it's of great importance to identify a number of other pathways associated with hypoxia-induced pathologies. So I have a smattering of them up here, including a number of thromboses, cardiac dysfunction, hypertension, and so on. So kind of understanding how other organisms have dealt with that on the genetic and molecular level might be able to help identify novel therapeutic avenues for hypoxia-related diseases. And, but on the kind of flip side of this is that hypoxia is not just an issue that organisms on land can encounter. Hypoxia is a phenomenon that can also be a naturally occurring part of the environment in both saltwater and freshwater environments, the most prominent of which are called hypoxia zones or dead zones. It's popped up recently in the news, at least it has in the past, that the more prominent of which, like I said, are hypoxia zones, especially in oceans. So what I'm showing you here is a map, kind of a widespread map, and each one of these red circles identifies a hypoxia zone that's occurred, and the biggest ones, at least for the US, are in the Gulf of Mexico as well as in the Pacific Northwest. So due to human-mediated climate change causing rising temperatures as well as pollution, effluent, so nutrient fertilizers, coming out through rivers and such, hypoxia than they normally would. They're getting bigger both in size and depth, as well as becoming more hypoxic. So they're getting worse in terms there's less oxygen available. So marine hypoxia adversely affects a wide variety of organisms, from fish to crustaceans, showing sometimes lower levels of tolerance, and this particular phenomenon can cause large economic losses in coastal fisheries and aquaculture, which is why some states are already trying to take action for the issue. So in terms of aquatic hypoxia, my general interest is then how is it affecting organisms living in those areas, and especially in terms of their distribution. So, okay. So I've kind of gotten through the general introduction of why I think hypoxia is cool. You may or may not be sold on that. But those are the big baselines and how it kind of frames the two major parts of the rest of my talk, which go over some of the work I did as a PhD student at the University of Miami with Dr. Kevin McCracken, and work I did as an NSF Fellow at Oregon State University with Dr. Felipe Barreto. So we'll get to talking about high-altitude duck species. All right. So I'm not sure how many of you are thinking about mountains. If you remember, the higher in elevation that you go, the less oxygen there is available, the thinner the air. So like I said before, at sea level, it's around 20-21% oxygen availability. The rest is nitrogen and other things. So I focused on animals that lived in the Andean mountains. They're actually the highest mountain range in the Americas, as well as the largest plateau outside of Tibet. They have a really extreme average height of around 4,000 meters. So on average, the percent oxygen availability is around 13%, which is pretty thin, with some places actually being around close to 7,000 meters. So the additional cool thing about the Andean mountains, at least in regards to the system that I worked on, the ducks, is that these plateaus have a number of different bodies of water, which are wetlands and marsh areas that a number of waterfowl actually live on, ducks and geese and even flamingos, as well as a number of human populations. I believe there's a city in Bolivia, which is the highest occupied human city space in the whole world, even including Tibet. So there's a number of animals which live here. And my PhD advisor, had been going to South America for 10 to 15 years at the time that I knew him, and when I started in his lab, collecting all sorts of samples, as well as general information about a bunch of different waterfowl species. So I'm showing you a smattering of them here and where they were collected. I didn't actually go out to the field. My advisor was the one that got mountain sickness. I just stayed on the computer. So I'm showing you, some of them have really cool color patterns. My favorite are these torrent ducks. There was a PhD student that worked on them. This is the male, and this is the female, and then these little babies are so adorable. So not only do they have to contend with high altitude, they actually are really active divers, swimming around and diving in high altitude streams. But I didn't do my PhD on anything nearly as exciting. I did them on what I called the duckiest looking ducks. So this is the yellow-billed pintail and the speckled teal. So the next little couple of vignettes about them will revolve around these two particular species. So I chose them based on all of the populations available because they independently invaded high altitude locations from one another, and they have both low altitude and high altitude populations that you can compare. And I wanted to do some work on them, mostly as it related to their genome. So what I ended up having to do, or what I was tasked with, was because we didn't have the resources available to just sequence their entire genome of all 60 individuals, my advisor was like, well, read through the literature and come up with a comprehensive list of a number of genes based on what we know previously that might be potentially under selection or associated with high altitude adaptation in the ducks. So that's what I did. I read through a number of articles and created a really comprehensive Excel sheet list with the reasons why I chose them. And the one that I found at the time, and continues to be a major one, is, so the big question was, do you know of any mechanisms for high altitude adaptation? And this might be the point in which some people's eyes glaze over, but don't worry about it. Oh, that's like really light up here, but you can probably see it better on the other screen. So this is supposed to be kind of an outline of a cell, and then you have the nucleus here. So the biggest mechanism was that of the hypoxia inducible factor pathway, and I'll call that the HIF pathway. Okay, so it's comprised of these two transcription factors. So those are the proteins that help DNA go from DNA to RNA, which then into protein, okay, as well as some repression machinery. So under normoxic situations, so just like sitting around, say like at sea level or whatever, where there's not that big of an issue, what actually happens is this transcription factors are basically tagged by this repression machinery for degradation. So they're tooling around, and then this enzyme complex comes, attaches things to them, and then other enzymes come by and then break them all up. So they're not able to do anything. But under hypoxia, when it becomes more and more of an issue, it causes actually a chemical reaction to occur within the blood that causes the repression machinery not to work anymore, which means that then these transcription factor members, alpha and beta, are able to move into the nucleus, attach onto the DNA, and then affect change through transcription, so RNA, which then eventually gets translated into protein and amino acids into protein. So this is just a smattering of some of the main things that the HIF pathway is associated with, including changes to blood levels, blood pressure levels, as well as production and change in angiogenesis and vascularization. So this we know actually a whole heck of a lot about the hypoxia inducible factor pathway, and that is partially in thanks to the labs of these three gentlemen here who won the Nobel Prize in 2019 for figuring out this particular pathway. And it's of interest mainly because of tumor hypoxia. So tumors create their own hypoxic environments. And so they were able to elucidate that a little bit more. However, I obviously used all that previous information for my own goals, which was to compare high and low altitude populations of these duck species and see which of the HIF pathway genes were associated with high altitude adaptation, if any. Didn't necessarily say that they would, but it seemed like it was possible. So I wanted to know if there was convergence on this pathway between these duck species. So I have this here, and I'll populate it with the actual data itself, but let me walk you through what the figure is supposed to tell you. So each species is on either side, and then you have numbers that increase in size, and the higher the number, means that more likely that particular variant in that gene is associated with high altitude, okay? And the rest would be low and that they wouldn't be. And I have on this y-axis just each of the genes that I chose from the HIF pathway. All right, so it might be easier to see on the other screens, but I think it pops out fairly obviously which ones have the most significant, and that is this EPAS1, HIF2 alpha, or EGLN. And if that kind of sounds familiar based on what you saw on the previous slide, it was because these were the exact same genes that are the transcription factor as well as member of their repression machinery. And what was kind of cool about that, in terms of the literature that I was looking at, is that human populations, especially Tibetan and Indian, those exact same genes were found to be under selection for their high altitude populations compared to their low altitude counterparts. And this has been shown, excuse me, time and time again in other systems like in yaks and Tibetan wolves and so on. Okay, hopefully people's eyes haven't glazed over too much, but ultimately what that ended up showing is that I found variants at this particular stage, right, at the DNA stage. But then I wanted to know if those variants actually affect a change from the RNA to amino acid to protein, because if they don't, then they're not as important and I can't really say much about them. Okay. So what I'm showing you here is a protein model, and proteins actually have very specific domain, so areas that have specific functions associated with them, and I won't go over each of them, but I figured since I was working with a system that wasn't as easily malleable in the lab, so I couldn't, bring in a whole bunch of South American ducks to Florida and try and experiment on them in the way that would be required, that if the variants fell between any of these areas, that it might give me a better idea in terms of like what it was doing functionally. So I'm about to show you what the variants were and where they fell. Okay, so both the speckled teal and the yellow-billed pintail, some of those variants were found in regions that actually got transcribed into RNA and then ostensibly translated into protein. And they, especially in this area called exon 12, sorry. And although not in the exact same area, they did show that there was convergence not only on the same gene, but in the same region of the gene. And what was kind of cool is it was in this area that is between functions, so both regulating oxygen tension, so when it's able to go into the nucleus and affect change, as well as a region that's known to be, remember when I mentioned the repression machinery comes in and attaches to stuff and then it's able to be degraded? It's in that same kind of area with two different functions. So that was pretty exciting to me. So I went further into literature to see if I could find information that showed anything about this, again, possibility of it being functional. So I was looking for other stuff that talked about this HIF2 alpha or EPAS1 exon 12. And by stroke of luck or otherwise, I guess, doing a really thorough job reading, I found that a couple of different examples where variation found in this exact same region causes a functional effect. So one of which is increased pulmonary hypertension. So it causes some elements of vascular change as well as cardiac function in cattle that are housed in Colorado. It's sometimes called brisket disease, which is kind of a sick joke, honestly. But so this lends credence to the idea that having variation in there actually causes additional downstream effects. Another example that I found was, although not necessarily high altitude related, that variation found in, again, the exact same exon was associated with familial erythrocytosis and polycythemia. So that means just an overabundance of red blood cells, which can be kind of problematic over the long term. So again, without being able to kind of do any work on, bring the ducks in and kind of crispring a duck or whatever, this led me to kind of form this hypothesis about what the variants I found are actually doing and how it's affecting a change and potentially providing an adaptive benefit for high-altitude the ducks. So ultimately, the idea is that somehow it's affecting a change through erythropoietin here, which goes into the bone marrow and changes blood cell production. So for me, the kind of next step was, what about blood? If that's my next kind of hypothesis. Just as a reminder, red blood cells are comprised of 4 different chains or hemoglobin to create this hemoglobin molecule, 2 different beta chains and 2 alpha chains, as well as a heme group where that's where the oxygen gets bound to. And these particular chains are transcribed or housed in these complexes, this alpha globin complex and beta globin complex. So what's kind of interesting about these complexes is that they house different versions of those genes, and they're transcribed or and then translated kind of depending on developmental time. So very early in development, you have some combination of them embryonically, and then there's a version of it briefly during fetal development before birth or hatching, as well as there's a kind of a major amino isoform with us that are currently circulating. So there had actually been quite a bit of work done both in humans and in the duck species on the adult versions. So I was especially interested in what was going on with the fetal hemoglobin. So as I kind of previously mentioned, as you go up in altitude, there's a reduction in fetal survival as well as fetal growth. And even when you take chickens and ducks from low altitude to try and breed them and house them at high altitude, there's decreased hatching viability. So I wanted to figure out if hemoglobin variation, especially in the embryonic and fetal versions, were associated with adaptation to high altitude environments. So what I'm showing you here, it hopefully looks a little better, it's a little washed out here, again, is kind of like a protein model in a way with a different kind of coding region, so an exon 1, 2, and 3. And I'll, again, highlight the areas which showed significant associative variation between their high and low altitude populations. And this is only actually in epsilon, so this is the beta version. I didn't find anything in the alpha. So what you'll notice right off the bat is that they have seemingly some amount of variance both at the same region, so both at amino acid 14, 15, 17, and 126. So that to me stood out and was kind of amazing that they would be at the exact same locations. But then looking further, notice that it didn't matter if I was looking at speckled teal or yellow-billed pintail, that the low-altitude populations had the same amino acid changes at each location. So at position 14, low-altitude populations had a serine, high-altitude had a glycine. So this was kind of a curiosity also in combination with what we knew about the adult hemoglobin, beta globin, that this might be 2 different things. There might be 2 different potential scenarios. So we know that the speckled teal actually invaded high-altitude locations before yellow-billed pintail. So one scenario is that there's just variation that occurred through random mutation for both of them. So you have your high-altitude population from speckled teal invade. They, over time, have adaptive variants which arrive and get swept to fixation. Later, you have your yellow-billed pintail invade. They similarly have variants that accumulate and are swept to fixation, and that they only look identical just a sheer chance, right? So that's one option. The other option is that you end up having the selection or variation being acquired through interbreeding, or gene flow, it's sometimes called. So in this scenario, again, you have your speckled teal invading high-altitude locations, acquiring variants, yellow-billed pintail invading, but instead of doing their own work, they interbred with high-altitude populations of speckled teal and acquire those variations. Sometimes this happens a lot in ducks. It's called the Desperation Hypothesis, because then there's not that many of them up there, and they really want to mate, and so they end up doing so with something that looks very similar. So it basically did, it kind of stole this very variant from the other high-altitude population. So I did a number of statistical tests to see whether or not which scenario was the case. And I'm showing you a number of those HIF pathway genes again here, as well as the hemoglobin variants. And the only 2 that came out as being significant were, again, those the adult as well as the fetal hemoglobin. So this kind of phenomenon, which is called adaptive introgression, is becoming something that happens more and more in the literature that we're finding. And some more prominent examples are variants in the immune system, metabolism, pigmentation, as well as high-altitude adaptation in human population, modern-day human populations from ancient Denisovans or Neanderthals. Maybe you've seen that talked about in the news. But there's also examples of, in butterflies, trading variations associated with predator defense, beak shape between different species of Darwin's finches, resistance to poison in different species of mosquitoes, as well as different species of rats or rodents, and not to forget about plants, a number of examples of herbivore resistance between really distant related species and genera and plants. Okay, so I just said a lot of stuff to you that may or may not have really stuck, but I wanted to give you kind of a log line. What should we have learned from these two little vignettes that I talked about? First, that this HIF pathway is a hotspot that I found both in terms of my duck species compared to humans, but continuously throughout the literature we find that these particular targets tend to be very predictable given their spot and how, because they're major players in this regulation of a very specific pathway. I also found convergence on the HIF pathway, not only on the pathway itself, but on specific genes and on the specific exon, which I think is really wild and exciting because it could have been nothing, which would have been really boring, but still would have been a result. And I found additional information of potential functional effects on hemoglobin production through that the variants were acquired through adaptive introgression. So just like any science, that's not where I stopped in terms of that I've answered everything. So I'm continuing to work on projects with my PhD advisor and other collaborators to kind of ask questions to test my hypothesis out about if erythropoietin is actually changing, is at the center of altitude adaptation, and if their expression is variation associated with it. And also looking at whether or not these hemoglobin variants actually change oxygen binding affinity as well, and if there's different expression levels in development. So those are kind of like pending potential projects. Okay, so that was all work that I did as a PhD student at the University of Miami. But in going through, you notice a few times I said in my talk, well, I couldn't do this, so I had to kind of rely on this previous information. So I was kind of feeling bound by the system that I was working in, which was attractive for some reasons, but was problematic for others. And I really wanted to kind of do more experimental work because I was running a lot of computational models and so on, and I wanted to be able to test them out. So when I was applying for a postdoctoral program, postdoctoral research positions, I eventually ended up applying to most places where they was using invertebrate systems because they're a lot easier to kind of bring into the lab and do molecular probing with. So I'll give you a few vignettes in terms of the work that I did associated with aquatic invertebrates, so copepods and other crustaceans, but going from terrestrial hypoxia to aquatic hypoxia. Okay. So the lab that I worked in used this copepod called Tigriopus. So it's called Tigriopus californicus because that particular species actually lives along, and this is really washed out, it lives all along the west coast here, but it does include populations up in Alaska and further down into Mexico and so on. They're very widespread intertidal copepod. So I have a picture here of me and some of my lab mates going out to the field and collecting some of them, and this is them in kind of a jug, and this is just one. They live in these really rocky shores, so you can kind of see it here, these kind of volcanic rocky shores in these ephemeral pools. So the pools themselves don't actually connect to the ocean at all. They only get water and nutrients and such from either the rain or from ocean spray. So you can imagine this creates a really dynamic environmental condition in which there's a lot of changing temperatures throughout the day, if not throughout the entirety of the year, as well as salinity changes, and then oxygen. So most of the work in this system when I came in had been done on kind of more low-hanging fruit. So this is like changing temperature and salinity in the lab, but I wanted to ask additional questions about oxygen availability. So thankfully my postdoc advisor allowed me to explore those questions in his lab with his consent and all of his materials. So here's just a picture, or let's see, will it let me? It should be playing. Oh, it's not playing. But anyway, so there should be a little video that's playing here, but all this orange stuff in here, those are individual copepods that are swimming all around and that are just, there's nothing really that eats them at all, and so they're really abundant. So you can just go out there, scoop them up with a small net, put them in a container, bring them back to the lab, house them and keep them and do work on them. And if the population crashes, you just go out to the nice coast again and collect them. So I did some field work for my postdoc here. So again, and why I spent so much time early on talking about this particular pathway is because it comes up numerous times. If you, just to remind you, remember HIF alpha is part of that transcription factor machinery, and EGLN here is part of that repression machinery. They constitute pretty much the major machinery associated with response to oxygen changes, both on kind of a widespread level, but also very tissue-specific level, because you can have certain areas in your body which are more hypoxic than others. And we know that they affect a number of downstream changes in the nucleus as well as in the mitochondria. And based on previous work that I've done, as well as work that other people have done, have shown that this pathway is really highly conserved, meaning that once it was acquired and had evolved within systems, it was kept. And so you can imagine, since oxygen levels, you want to keep tabs on that, and since all animals, for the most part, use oxygen as a key limiting factor, you'd imagine that pretty much everything has it, and that was what the literature had suggested up until that point. And again, they were a common target for selection. So initially going into my postdoc position, I just wanted to kind of get a handle on the members of this HIF pathway and what they were doing, very generally because my assumption was that it would definitely be associated with response to hypoxia in these copepods. So my initial goal was just to search and identify the presence of this HIF pathway in the copepod system. So it wasn't initially testing any questions, it's just like keeping tabs on it. But this led me down the rabbit hole because I ended up finding something kind of quite unexpected and made me shift my research questions from that point forward. So what I'm showing you here is instead of, say, like a phylogenetic tree, which has different species that it's comparing, I'm using phylogenetics to assess gene family membership. So don't worry too much about all these individual branches, but you'll notice that there's these gene families that are labeled along the base here, and that I have a number of known control species of different colors, and that all the different populations of the same species of copepod I was working on are in blue. So with each of these families, you'll notice there's some contingent of blue in every one of them, but do we see any evidence of gene membership associated with HIF alpha? So that's the one off to the right. So you'll see that there's no blue here, when all previous literature had said that everything has HIF alpha and EGLN, except for I detected no presence of HIF alpha or EGLN. So this is another trait that I didn't include. So at the same time, also to test as a kind of side way of testing to see what the HIF pathway might still be doing, or if it's just available in portions or if it's completely gone, that there was no enrichment of these elements at which these transcription factors bind to. They're called hypoxia response elements. There was no more, basically, that these hypoxia response elements had degraded over time because there was no more need for them, basically. So there was no transcription at all. So it seemed like this HIF pathway is completely obliterated and is not being used in this copepod system. And at the time, this was the first time the HIF pathway loss had been documented. So, this was pretty exciting and daunting for me, which is not what my expectation was going in, but kind of let me the ability and the option to pivot and kind of really think about how I wanted to use that new information and move forward with it. So we know that it was missing the HIF pathway. Does that mean that they can't really respond to low oxygen very well? So what I ended up doing is taking copepods and exposing them to, in kind of this jerry-rig situation, different oxygen levels. So what I'm showing you here are the survivability of just normoxia controls. So I wanted to make sure that the tubes and stuff I was putting them in wasn't killing them. And then what I did is I exposed them, I pushed them to their limit, which is near anoxic exposure, so less than it's very, very little oxygen left for a number of days, 24 hours all the way to 96 hours. So what I ended up finding is, obviously, after a certain period of time, they don't do very well, but even at 24 hours of basically no oxygen, there was no significant difference in terms of their survivability. They seemed happy as clams, as it were. Then they started seeing kind of like successive die-off, and one measure of this over a period of time is called LD50, so it's like time to death. About 50% of them had died, and this was at 52 hours. And comparing other crustaceans, this is actually on par, if not better, than a number of crustaceans that had been tested previously. So not only have they missing their HIF pathway, they're doing just fine without it. So I think that was wild to me and to my advisor as well. So what I ended up doing is, since we could keep them in the lab, they carry these little clutches on their legs. They carry them. So you can pick them, pick the clutches and put the females back in. And once you pick these clutches, after a certain period of time, they hatch. I wanted to control for their age, because obviously the older they are, the more likely they're not going to do nearly as well. So I grew them up so they were all around adulthood of 20 days, and then exposed them to either anoxia or this normoxia control experiment for either a short period of time, a longer period of time, which I knew bordered on the line between it being detrimental, as well as exposing them and then giving them 24 hours to recover. And so ultimately what I'm doing here is looking at this DNA, this RNA portion. So I sequenced, it's called RNA-seq, and it sequences all the RNA that's available. And then I was comparing this normoxia to anoxia control. All right, so I'll give you very generally kind of like the big takeaway pictures. So at 3 hours, only 26 genes had a significant difference of expression, which if you think in terms of, in humans there's something like 20,000 genes, in Tigriopas there's something like 16 or 17,000 genes, that only 26 genes really seem to have a response, which is incredibly low. So, and I noticed even at 3 hours they were more than happy swimming around. They didn't really seem to be bothered by it whatsoever. So it's a really tight transcriptional response, but the genes that did change were those associated with transporter activity, so things like sodium, potassium pumps. And it wasn't until 24 hours that it seemed like that the transcriptional landscape actually took, knowledge of the fact that there was something happening. Again, there was a response expansion. So of the 15 genes, 11 of those genes stayed on. So 42% of these 3-hour genes stayed on at 24 hours. And then a wide variety of these genes here were those associated with metabolic activity in the mitochondrial matrix. So that kind of makes sense given the role of mitochondria in producing energy associated with oxygen. So you go from oxidative phosphorylation into glycolysis. And then at 24 hours, where they had been exposed for 24 hours and then left to recover for 24 hours, there was very little overlap between this 24-hour anoxia as well as the recovery, only 52 of those genes. And that any overlap was due to complete reversal of expression. So if it was upregulated, so meaning there was more expression of RNAs at 24, there was a complete reversal, so it was downregulated at recovery. And a lot of those genes are associated with muscle structure, development, and motor activity. So genes associated with the extracuticular structure of the organism itself. Okay. So, even though I was able to get a slight piece of information associated with what was happening, ultimately, the layout is, the 3 men that won the Nobel Prize, we had this all figured out. The whole literature was set up to where this was completely understood. And now we have so many question marks, which can be particularly daunting, but I think opens up so many opportunities to really figure out what are they using instead, if they're using anything at all, as a kind of a weird example. So that led me then to wonder if this HIF pathway loss is actually idiosyncratic in Tigriopas. So meaning, are they the odd ones out? Or does it mean that there's other crustaceans that have a similar issue, but that they're also missing it? We just took them for granted in terms of looking at it. So I looked at Tigriopas are crustaceans, which are part of this larger group, and arthropods, which insects are in. So what I did was look at a variety of different crustacean groups. So I'm showing you here is a whole bunch of the major ones, including, shrimp and crabs and amphipods. You have your barnacles, which are technically crustaceans, and then as an outgroup, I have the insects. So I used a whole bunch of genome and transcriptome data that's available online. Again, checked for the presence in the same way that I did in a previous figure with those phylogenetic trees. of these major areas, the repression machinery, as well as the transcription factor alpha. And again, the goal was to search and identify for the presence of HIF pathway, but in other crustaceans. So I'm going to show you, again, a figure that comprises or summarizes a whole bunch of work and information, which ultimately, a wide variety, obviously, of crustaceans do have HIF alpha and EGLN. However, I noticed that there were several additional losses in copepods, as well as a completely unidentified loss within barnacles, Cirripedia. So I wanted to zoom in a little bit further, and what I'm showing you here is the phylogenetic tree known in copepods. There's a lot of question marks because there's not a lot of genomic resources available, but it wasn't all copepods that had lost it since calanoids had the machinery. But at some point during their evolutionary history, there was a loss, and that included cyclopoids, harpacticoids, which Tigriopus is a part of, as well as cyphostomatids. So cyphostomatids are a parasitic member, and this is an example of their ectoparasites. And this is one on a Greenland shark. It's like on its eyeball. It's pretty gross, but they're a major issue for a lot of commercial fisheries like salmon and other fish that attach onto them. And at the same time, looking at barnacles, there's even less information about them available, but that it seemed like the loss was whole scale within the entirety of barnacles. So again, both Cecilia, Pendicula, so your kind of very general barnacles I'm sure you've seen before, but also they have their own kind of parasitic lineage, Chondrogonadids. So this is actually a barnacle, this thing that's moving. So what it does is it parasitizes other crustaceans and kind of mind controls them by infecting their brood pouch, and so that the crustacean takes care of it, but it's not actually their baby, so it's pretty sad. But it's also really wild. So there's a wide variety of like cool, interesting things. So what is the overall, what have we learned for kind of this set of vignettes here? So ultimately, there's no HIF pathway, and that they're potentially using alternative mechanisms, but we really don't know anything about it. Some information initially is that potentially they're using exoskeletal modifications, they're changing their metabolism, but we don't really know anything about what their transcription factor machinery is, if they've co-opted something else, or if they just were fine without it. And that this HIF pathway loss is not unique to Tigriopus, that I found evidence of loss in barnacles as well as some other copepod groups. And then again, these losses are independent of one another, but there's some information to suggest that copepods and barnacles are maybe more related. That's not entirely known, but there may be something about their physiology and their oxygen needs which allows for this loss to occur. So again, what I found didn't just create a wall and then there's no more questions to ask. There's additional questions. So what are these transcription factor machinery that they're using? And what I didn't show is that there's multiple populations clearly of Tigriopus, but they all had slightly different response levels, so they didn't all survive in the same way. So I don't know whether or not they're using similar mechanisms. The same thing goes for barnacles. So what are they using instead? So there's just opened up the possibilities, and it has the potential to be daunting, but I think is really exciting. So I'll kind of end the science portion of that by also mentioning that I don't do, just science out there, and maybe there's this unintentional emphasis on that all a scientist is doing is just research and publishing and presenting. But I've found it really important, especially from my unconventional background, to do a lot of outreach and broadening participation. And so I've been really active in hosting elements of Girl Scout badge days, participating in starting these lab workshops, and even here at the University of Utah, this Genomics Summer Research for Minority Group program that happens every summer. So if that's something you guys are interested in, you should check it out. It's like an 8-week course or 8-week situation that's paid, and you end up getting housed in a lab and doing research with a project. So I think this is very important. I've been doing it for a long time and was actually part of my fellowship at the NSF. So I'm continuing to be passionate about that going forward. So hopefully I didn't take up an incredible amount of time, but I really like to thank, of course, all the people that I relied on over the course of my training, all the numerous societies and foundations that have helped pay for my research. And even though I didn't really get to go to any field sites during my PhD, here's a smattering of the beautiful coast along Oregon, and I was able to go to Arches recently, so I have some pictures here. So I'll take any questions if there are any now, and thank you guys so much for letting me talk about my science.
Thank you, Dr. Graham. I realize that it's time for you, some of you need to leave to go to another class. If you'd like to come forward, any of you who have questions. On this side, if you came in this door that's on the south, please check with the TAs. There was something wrong with the scanner today, so please check with the TAs to make sure that your attendance got checked if you came in on the south door. Thanks, guys.